This is a continuation of the session from before lunch on mechanisms of interaction. And we have two more talks in this session, and then there is certainly some more time for discussion and then a coffee break after that. Our first speaker is Professor Vicki from the Heriot University in Edinburgh. And the title of the talk is Impact of Route of Exposure on the Systemic Effects of Nanomaterials.
Thank you very much. I'm going to show you today some results from a few studies that we've been involved in recently, and I've tried to combine them together so you can compare and contrast the results to allow you to identify whether you think the in vitro models are any good or not. So we can take particles into our body via various different routes, and the work from Wolfgang Kreiling's research group that was based in Munich elegantly showed that particles that are inhaled and some more recent work after ingestion, some of which hasn't been published yet, clearly demonstrated that a small percentage of the administered dose, so maybe just 1 or 2% of the administered dose, could cross the barrier at the point of exposure and gain access to the cardiovascular system. But then of that 1 or 2% that gets into the blood, more than 95% of the particles accumulated in the liver. So a lot of the work that I'm going to show you today focuses on the liver as a potential target for nanomaterial toxicity. One of our challenges in this research field is to promote and improve the use of in vitro techniques because we have too many nanomaterials to test in vivo. We've seen a lot of in vitro data today, and there have been questions already about the usefulness of some of those models and how they reflect the in vivo scenario. And we need much more information about that in order to allow extrapolation and full use of the in vitro data for risk assessment purposes. So the first study I'm going to describe today is part of a project which is now finished called In Live Tox, an FP7 funded project. And in this project we used 3 particle types, a gold capped with PVP and a TiO2 and a silver particle. They were fully characterized, or I don't think the word fully is right, they were extensively characterized. And they were used for in vivo studies in which they were administered to rodents either via gavage, so the oral route of exposure, and as a comparison, IV injection. And the IV injected dose was calculated on the basis of how much we knew translocated from the gut into the cardiovascular system from the biokinetic studies that were carried out prior to the work that I'm showing you. We sampled the intestine, the Peyer's patches, the liver and the aorta, and we measured oxidative stress, inflammation markers, cytotoxicity markers, apoptosis markers, and functional markers for each of the tissue types. We then took some representative in vitro models for each of those organ or tissue types. So there was a CACO2 cell line for the gut, human umbilical vein endothelial cells for the blood vessel endothelium, and the C3a hepatoma cell line for the liver. And we measured the same endpoints using the same assays as far as possible in the in vitro system as we did in the in vivo system, and they were measured by the same person in the same lab. This project, one thing I should say, was also conducted alongside the FP7 project Emperor, and we were also working on some of the same particles that I'm showing you here in that project, some of the same cells and some of the same protocols. So those protocols were standardized and used across those different projects, and I'll show you some of that data in a moment. So this is the liver cell in vitro, and this is a marker of oxidative stress. So we measured the glutathione content of these cells to indicate whether they was depleted and and therefore an oxidative stress. And we found that with increasing concentration, there was a significant depletion of the glutathione content of the hepatocytes in vitro with some of the particles, such as titanium dioxide. If you look at the same particle, titanium dioxide, in vivo, we were also able to demonstrate a depletion of glutathione in the liver after an IV injection. So here we're comparing an in vitro hepatocyte cell line where particles are dispersed in a medium containing serum via a very strict protocol that was shared between Emperor and in-life tox to disperse the particles. The same dispersion protocol, or similar dispersion protocol, was used for the in vivo situation, but here we have an IV injection, and you see comparable data. We then looked at gene expression. In vitro, we looked at gene expression for IL-8, which was upregulated at the lowest concentration and to a smaller extent at the higher concentrations. We also saw an upregulation of IL-1 receptor in the C3a cells and an upregulation of TNF-alpha. Note the dose dependency of those upregulations. If you don't look at low enough doses in your in vitro system, you can measure these, you can miss these responses. The dose is expressed as micrograms per centimetre squared to give you the worst case scenario dose. So that assumes that 100% of the particles in your system are settling down onto the cells. That's probably not true, but it gives you an indication of the worst case scenario for the dose. This is the in vivo data after an IV injection. All of the genes which were upregulated in terms of pro-inflammatory responses in vitro were also upregulated in vivo with this silver nanoparticle. This is now in vivo data. On the left-hand side is after an IV exposure, and on the right-hand side is after a gavage exposure. So I haven't got in vitro versus in vivo here. I've got two different routes of exposure in vivo as a comparison. And you need to look at this first column here. Yellow means no change. So after a gavage exposure, we saw no change in the IL-1 receptor and the MIP2 gene after gavage. But we did see those upregulations after an IV exposure. In both exposures, we saw an upregulation in TNF-alpha shown in green here. So what that tells you is that the route of exposure determines the impact on the liver. So once the nanomaterials go in via gavage, their impact on the liver is different to when it goes in via an intravenous injection. And that the in vitro model is good for the in comparison to the IV route, but the in vitro model is not good in comparison to the gavage route of exposure. So that's just expressed there. So either the passage through the gut has either given the particles some kind of different property, maybe some coating that they've picked up on the way, or the particles have impacted on cells in the gut, which have sent a humoral or chemical or neural signal which has influenced the liver response. The parallel study, which I mentioned, NPRO, using the same cell type, same dispersion, the same particles, the same protocols for measuring cytotoxicity, etc., looked at 10 different nanoparticles. I'm just going to show you the data for two of them, silver and titanium dioxide. So the bars represent cytotoxicity. So you see, sorry, the line represents cytotoxicity. So you see a dose-dependent increase in cytotoxicity with the silver and less so with the TiO2. And as the toxicity increases with silver, you see an increase in IL-8 production shown by the bars, but then it drops off as cell death advances and the cells become less able to produce the IL-8 protein. In contrast, the TiO2 never reaches sufficient toxicity to inhibit that IL-8 production. We compared the C3a cell line in that study with primary human hepatocytes, and we compared 6 different particles, and we saw very comparable results between the primary hepatocytes and the C3a cell line in terms of the impact on cytotoxicity of these particles on the hepatocytes, suggesting that the cell line is quite a good model for primary human hepatocytes, and that cytochrome P450s, which are not highly expressed in the C3a line, are therefore unlikely to be involved in the hepatocyte response to particle exposure. This work was conducted at the RIVM and is the inflammatory response that occurs in the lung this time. So the route of exposure now is an instillation into the lung for the same two particle types, silver and TiO2. The silver did not induce an inflammatory response via instillation, but the positively charged titanium dioxide particles did. We also measured the glutathione content of the liver from these animals and found that we saw, I was quite surprised by this, a significant depletion of glutathione in the livers of these animals following a silver exposure and a TiO2. So remember that the silver did not induce a lung inflammation. There was no measurable lung toxicity or lung inflammation in these animals, yet they were still able to induce oxidative stress in the liver. We also measured glutathione content in vitro, and both of the particle types, the silver induced a significant depletion of glutathione, but the titanium dioxide didn't. So this is the in vitro model here is not necessarily reflecting the in vivo response. In fact, we looked at 10 different particles, and we found that for 7 particles, the cell line could predict the in vivo liver oxidative stress response, but for 3 of the particles, it could not when they were administered via the lung. The particles which did not reflect well in vitro compared to the in vivo model were the multi-walled carbon nanotubes, and we think that might be just something to do with the large agglomerate size of these particles, maybe affecting how they behave in the lung and whether they can cross the lung barrier. And the positively charged TiO2, the positive charge can enhance cell penetration and barrier penetration by particles, and so in vivo, we're seeing maybe a bigger response than you might have expected. So the comparisons are then for serum dispersed nanomaterials, the hepatocyte cell line seems to be a good model for predicting hepatocyte responses to nanomaterials in terms of uptake, cytotoxicity, and inflammatory responses, and therefore cytochrome P450 seem to be not involved in the responses that we measured. If you compare the in vitro versus the in vivo liver responses following a pulmonary instillation, the particles which did not induce lung inflammation could still have an impact on the liver, first of all, and the glutathione depletion in the liver of the mice reflected the in vitro responses for 7 out of 10 of the particles. We've also looked at how the corona, or the particle coating, can be modified by our culture conditions. Typically, as I said in the for those previous studies, people disperse their particles in a medium containing serum, but we don't really have pure serum in the body. We have plasma in the body. So we compared the corona composition for 10% plasma with 10% serum. There are some similarities, but as you can see, there are also some differences. And then we thought, well, if you inhale a particle, the particle doesn't instantly become covered in either plasma or serum. It becomes coated in lung lining fluid. And then if it does translocate from the lung into the blood, it will then interact with serum components. So what would the corona look like in that instance? So we suspended the particles in lung lining fluid, centrifuged them until there was a stable corona on the surface, and then suspended them in serum or plasma. And this work was conducted in collaboration with UCD. And in that instance, you completely change the corona. There are some similarities, so there are some complement, albumin and apolipoproteins. But if you look in particular at the lung lining fluid followed by the serum, you start to see things like IgG or IgC accumulating on the particle surface at relatively high amounts. We also looked at the chemotactic response to particles after they've been coated in different, either plasma at different concentrations or serum or lung lining fluid. And you can see that if you change the coating on the surface of the particles, you influence their ability of these particles to induce chemotactic responses by macrophages in vitro. We also looked at macrophage and hepatocyte responses in terms of pro-inflammatory gene expression. Here we've got medium gold of 2 different sizes suspended in medium, gold of 2 different sizes suspended in lung lining fluid, and then exposed to hepatocytes. And then we measured a range of different cytokines, so IL-6, MCP1, IL-10, TNF-alpha, and IL-1 beta. And the pattern shows you the kind of distribution of cytokines that were made in response to those different treatments. So you see differences between particle size, but you see differences between particles distributed in medium versus lung lining fluid. The lung lining fluid for the hepatocytes seemed to enhance the pro-inflammatory response, but the lung lining fluid for macrophages did not enhance the pro-inflammatory response. So in summary, hepatocyte cell lines and serum dispersed nanomaterials. They compare well with primary human hepatocytes in terms of cytotoxic and pro-inflammatory responses for 10 different nanomaterials. They compare well with the rat liver responses following nanomaterial injection in terms of oxidative stress and pro-inflammatory responses for 5 nanomaterials. They compare well with the livers of pulmonary instilled mice for some nanomaterials, but not for others in terms of oxidative stress responses. But they do not compare well with the liver responses following gavage exposure to the particles that we investigated. The responses of macrophages and hepatocytes and liver tissue is nanomaterial specific, it's corona composition specific, and it's route of exposure specific. So we have a very complex situation on our hands in terms of trying to make this simple in the future. I'd like to thank my colleagues at Heriot University and all of the collaborators that were involved in the work today. And before I finish, I'd just like to say that for all of you, your input into this project ITS Nano, which is about developing an intelligent testing strategy, is very, very welcome. And this document, this strategy, will be circulated to many of you in the near future, in the coming weeks, I hope, and your feedback is very, very important for this project and for the European Commission. And for those of you who wish to attend a workshop to discuss this strategy in more detail, then please just contact me.
Thank you very much. This presentation is open for questions. Perhaps if I may start with one question. So you mentioned that hepatic responses to nanomaterials are unlikely to be explained by the cyclone P450s. And in fact, and as you also, I think, alluded to, it's quite difficult to maintain hepatocyte cell lines and with a normal repertoire of cyclone P450 enzyme expression. But if one instead asks the question, I mean, does nanoparticle exposure in the case of primary hepatocytes change the expression of cytochrome P450s, which would then affect how these cells respond to other xenobiotics?
That's an interesting question, which we've not investigated, and I think it's highly possible, especially if oxidative stress is involved in that, and I think P450s can be modulated by that signaling pathway. So,
because we're probably rarely, if ever, exposed to only one single type of nanoparticle at a time. We're exposed to combinations of various offending agents and drugs and so forth. So it might be interesting to ask that question.
I agree, definitely.
Okay. Yes, there are some questions in the front row.
Yes, my question is, what's the half-life of the silver particles? Too. So it's quite reactive.
Okay, so I meant to mention the solubility of the particles, and I didn't do that. I apologize. So the silver solubility was measured, and it was less than 1% soluble in the conditions of the study conducted. The zinc oxide, if I mention zinc oxide today, in contrast, is more like between 40 and 60% soluble.
So the CO2 is quite comparable.
Yeah. I mean, we thought that the silver particle toxicity might be explained by silver ions, but the solubility was relatively low.
There was one more question. Kind of. Okay.
Well, just kind of, because part of it was already asked now. However, the question is, did you check what are all these responses with the dissolved ion? Because even if it's only 1%, this can change with time first.
I don't have that data with me today. I need to check. So for some of the endpoints, we have done a dose response with soluble silver ions and with zinc ions, but not for all of the experiments. And the reason we didn't do it for the silver is because the solubility was so low. But it's a good question, and I think for the zinc oxide, I'm pretty confident that some of the responses we see are soluble ion mediated. I'm less confident that that's true for silver, although intracellularly there could be additional dissolution as discussed this morning.
Can I ask a last question maybe? You showed for some conditions also an upregulation of interleukin 10, which is an anti-inflammatory cytokine. So it's produced along with pro-inflammatory cytokines.
Yep.
What do we make out of that?
Okay, so I have asked an immunologist about this, and she said it's not unusual to see that, to see pro-inflammatory and anti-inflammatory cytokines upregulated at the same time, and the trick is to work out what the overall situation is there. And actually, we've just had a manuscript accepted where we did IV injections of particles and then looked at the liver histology over time, and we looked for neutrophil influx because IL-8 was very highly upregulated in all of the studies that we did, whether it was in vitro or in vivo. And you see significant neutrophil influx into the livers, but only for 24 hours. And after 24 hours, I think by 72 hours, they're gone. So it's a very transient response. So I think that's, as well as seeing a pro-inflammatory response to trigger your, as Bent said earlier, a response to help you clear the particles. It's a protective response. You need that. Afterwards, the liver seems to be recovering from the particles we've looked at.
I think the message here is that the cytokine profile alone is not going to be sufficient to tell you about the immune response. I fully agree with that.
Yes, of course. Fleming.
Fleming, make it an easy one. I'm not feeling very well.
Well, the study that we did there with the IT injection in the lung, where you don't see any effects in the lung, but still you see effects in the liver. Do you know any endogenous component that can cause these effects in the liver, or is it absolutely sure that it's the particles or even the soluble fraction of the particles have?
I have no idea whether it's due to particle translocation or whether it's some kind of soluble mediator or a neural response. I don't know the answer to that. And no, I don't know anything else that does that. It was a surprise.
Okay, so thank you very much.